learn that in the near future. So my course will be very brief, but hopefully you can get an idea how we can work together, pharmacists and the clinical laboratory. Okay, so I'm going to talk a little about laboratory tests to direct antimicrobial pharmacotherapy, which I'm not quite familiar with. I'm quite familiar with laboratory tests though. So, okay, so today we will do several parts. A little introduction. Talk about direct observation of the offending organisms or pathogens, and isolation of the offending organism. Other lab tests that can be very helpful actually, and more used nowadays. And another very important part for you guys I'm most interested in is the in vitro antimicrobial susceptibility test. And I'm also going to talk a little bit about monitoring the drug levels in your patient within the laboratory. So learning objectives for you guys to review after class. A little bit introduction. So the overall idea we're standing here is because we're taking care of patients, right? You, the physician, the care team, and us. So our patient is showing some history, signs and symptoms, and they present themselves to the clinician, right? And our journey starts here, diagnosis and treatment of infection. We want to make sure what they have, and then give us an idea how to treat them. And the patient themselves present the history, and they also show their symptoms, right? These are collected by the patient care team. And to present, they share the information with us as clinical laboratory. And what we do here is the highlighted part, but also hided part from you guys, from the physician, from the patient as well, is the isolation and identification of the pathogen. So what we do, we help you guys to know what is causing the infection, right? And a lot of organisms, you want to isolate them, make sure you have the pure culture, so that you can do what? To do the susceptibility test, especially for a bacteria, right? For those, you want to use your drug wisely, cautiously, and effectively. So susceptibility test is very important, and it's also carried out in the laboratory. So, clinical laboratory, I do have a video for you guys to review after class. That's 10 minutes from ASM. This is a career path that you can take. You still can take right now. You can change your mind and join us. Clinical laboratory, we have multiple sections. What we are going to talk about today is the microbiology section, right? We also have, if you're not familiar with, we have hematology, we have blood bank, we have chemistry. So what we do behind the scene is we perform the lab tests that helps the diagnosis of infection or other diseases, and we also investigate the microorganisms in the microlab that cause infectious disease. So by identifying those organisms and do the susceptibility test, hopefully we can provide useful information for clinicians and the pharmacists to treat your patients properly. So, to begin with is, how do I know my patient is having infectious disease, right? When they present themselves to the hospital, to the clinical clinics, they always show certain type of symptoms, right? Are you guys familiar with those? Can anyone volunteer say some symptoms? What can be a symptom of patient having infection? Fever. What else? Pain. What else? Yes, very good. So all of that symptoms gave us an idea, hey, there's something going on, it's very highly likely it's an infection, right? So the clinician will decide what test to make sure this is an infection, to make sure, hey, we are heading towards the right direction. So they're gonna order a bunch of non-specific tests, and these are kind of routine. Everybody gets those. So when you present yourself to a hospital, you're gonna get, if you're not familiar, you're gonna get Chem 5 or Chem 7. That's the chemical analyzed to determine your basic environment in your body, and you're gonna also get CBC, which is a whole blood count. So within that CBC, there's a very important criteria that is called white blood cell count. Okay, so way behind the scene, we use your sample, we make a smear, and we examine it under a microscope and decide, hey, what is the component of your white blood cells, right? Are you guys familiar with white blood cells? Learned immunology? Yes? So can you tell me, well, at least I labeled them, all right? So tell me which one is more important if you have a bacterial infection. Let's sit there. And here's something. Leukocyte, yes. Yeah. But all of these are leukocytes, if you know. Okay, so leukocytes are white blood cells. They're the same. Neutrophils are the ones specifically indicates bacterial infection. Okay, so when we do the CBC, we do the white blood cell count, we do the differential, we're gonna tell you which cell line actually increased, decreased, or have any abnormal cells within them. So I gave you a range here, a normal range of white blood cell. In overall, your overall leukocyte count should be this much, but I also tell you, it varies, depends on your patient, right? If you're a hospital, take care of infants or little kids, the normal range will be different. Take care of mainly women will be different with those take care of men or, old people. Anyway, so this is developed by your laboratory, but you always have a normal range with your patient. So if it's outside the normal range, it gives you an indication of something going on. So a lot of time, if you see a bacterial infection, you will see leukocytosis, meaning your overall absolute count of your white cell will be increased. Okay? And that is called leukocytosis. I do want you to remember the name, the definition of leukocytosis. And within that leukocytosis, you're going to see a lot of increase in your neutrophils and even left shift. Left shift means your neutrophils will become a little bit more immature than it's supposed to be. What does that mean? See here, far away. I'm going to use my pointer here, not that one. Right here, do you see this little guy with a band or a horseshoe-shaped nucleus in there? That's called banded neutrophil. So that's an immature neutrophil compared to this one, this guy right here. That's a mature neutrophil that's called segmented neutrophil. Okay? So if you have left shift, you will have more of the immature form of your neutrophils. And that's also associated with the bacterial infections. A lot others, I won't go into depth. Viral infection. So if you have viral infection like COVID-19 or something, HIV even, sometimes it will give you a normal or even decreased white blood cell count because a lot of the neutrophils kind of hide it out. And you will have, sometimes you will have an increased lymphocyte because lymphocyte is the major cell line that defends viral infection. Okay? And parasitic infection. What is parasitic infection? Give me an example. Hey, good, very good. Parasitic infection, sometimes you will see increased eosinophil cells in your overall white cell count. Eosinophil is this pretty one right here. It's not that pretty in here, but if you see, they're bright orange color and very pretty for us. Okay, other biomarkers for infection. You can also get tested for ESR, CRP. Oh, one indicator for my lecture is I like to underline the important things. If I have them underlined or in bold font, then that means kind of important. I want you to know, okay? And these are called acute phase reactants. So with these, they're non-specific. Everybody have any kind of inflammation going on, they may have increased value of these. But they are also very important for infectious disease because inflammation is definitely going to happen with your patient. So with ESR is a test that's going to test out speed your erythrocytes settle in the tube. So this is the little device for ESR. That's a total manual method. You also have semi-automated method, but overall they use the same mechanism. They detect how fast your red cell is going to settle. And the reason why it matters because if you are having inflammation, you will have increased serum protein. And this increased serum protein is going to decrease the zeta potential. You guys know zeta potential? That is a little negative charge around the red cell to make them repel each other. So if you have zeta potential, your red cells tend to avoid touching each other. So this increased serum protein is going to decrease that. So make your red cell easily, compose together or backward together, okay? So anyway, so if you have inflammation, you have infection, you will have a faster rate of ESR. C-reactive protein is another indicator quite often used today. That is actually more popular than ESR. ESR is kind of figured out. So this is measuring a liver synthesized protein that is really sensitive and responsive to inflammation. If you have any kind of inflammation, you will have a rise of CPR. And we have automated method and manual method, qualitative to detect like your elevation and how much exactly do you have in your body. One last thing I want to introduce about lab biomarker is the PCT. PCT is more specific related to bacterial infection. So when you have a bacterial infection, especially with someone who has sepsis going on, they tend to order PCT a lot of time, procalcitonin. So with this, it will give you an idea whether you should continue your antibiotic treatment, whether the patient is responsive to your treatment, or whether it's time that they've been cleared out and you can discontinue your treatment. I won't go into details about the testing method. I do want you guys to have a brief idea, like what we use to measure these biomarkers, right? So for PCT, we have manual test kits, also a semi-automated system to measure. Okay, so once I decide, hey, my patient's definitely going have some inflammation or infection going on, what I want to do next is to order lab tests to make sure what organism is causing the infection, right? So the most important thing is to, if I can see them directly, that would be perfect. So direct observation, and if I keep touching that. Before that, I want to clarify some information about pathogen and common flora. So pathogens are the ones that are capable of damaging you, causing disease, infect you, and produce all the signs and symptoms of infectious processes. So it includes all kinds of organisms, not only bacteria, fungi, viruses and parasites, all of them can be pathogens, right? There are also common flora all over you. We are not a sterilized person. We are colonized by all kinds of bacteria, fungi, and viruses and parasites as well. the common flora, the organisms can become pathogen if the patient has certain type of immune compromised situation, or they make a way cut through into your system, entering your bloodstream. They can cause, even they normally consider common flora, they can cause disease, right? So I listed a couple just to make my slides look better. Overall is that not all of the microorganisms would be considered pathogen, only the ones that's actually causing the disease. Okay, make sure that's in your hand. Okay, and to go with the laboratory processes, I think it's a little bit more clearer to see overall at the very beginning. So we collect samples. Hey, the infection is suspected. Then we collect the samples. We try to observe the microorganisms within the sample. Okay, so that's our next step, direct observation. And after that, we do want to make cultures and doing tests to make sure the organism we have on hand is the one that is the pathogen, right? And once we have the isolation, we have the culture, the pure culture, we can do the drug susceptibility. But this part is mainly talked about bacterial infection, not so much about viral matters. So the samples, I do want to show you a little bit more the process. Samples, our lab can get a whole bunch of samples, right? Blood culture, meaning you're gonna draw patient's blood. Once they're gonna draw into the blood culture tubes, meaning the physician is thinking, hey, they might have sepsis or they have infections in their bloodstream. Wounds is quite often, right? It could be localized, it could also go systematic, depending on the severity. And the wound will be collected into these sterilized tubes. CSF, do you know what is CSF?
Cerebral fluid.
Aw, so nice. Okay, I'm impressed. All right, so CSF is another very commonly seen sample in our lab. Oh, I do want to mention this little cute guy right here. Corner, that is the blood culture machine. What it does is to detect any color change that's caused by any growth in the blood culture. So even though there might be growth, it could be just common flora on your skin when they do the stick, or it could be something irrelevant. But anyway, this is very cute. I do want to show you that. And urine is another very common one that you're gonna get from the lab, in the lab. And sputum. I left one very important sample out. Can you guys think of what another very commonly gathered sample from the patient having infection? You guys are so smart. I'm touched. Okay, yes, I left the stool sample out, fecal sample out, because I do want to mention that during gram stain. All of these, we have certain standard of process. Depending on where we get a sample, we have different procedures to proceed with them. Take the bacterial infection as an example. Okay, so what we do normally is we are going to do morphological tests, which are the staining techniques, direct observation, and then we will strip the plates, or depending on what bacteria we're talking about, we might inoculate a graft, right? Try to make sure they grow, make an enrichment, and we pick a colony and strip other plates to let them show the cultural characteristics, because I'll talk about the media later on, that they will show different characteristics on different media. So that's a way to differentiate and identification as well. And the old microbiology labs, they do a lot of biochemical tests. That includes Indo test, urease test, catalase test, you can name it, go on and on. And after that, different organisms, they will show different characteristics of a reaction. Some will show positive for indole, negative for catalyst, and we just make them onto our chart and go through the chart and see which one they led us to, right? And nowadays, more and more, these traditional testing methods being replaced by immunological tests and molecular tests. And I'll talk about these later on. Okay, so a direct observation normally is done through a gram stain, okay? Unless you're certain there are something else, not a bacterial infection, then you may do other stain. But like 99% of the staining in the clinical lab is gram stain. Gram stain, how do you do that? Just a little bit introduction. So the staining begins with you fix your cells on the slides, okay? Your patient sample, you grab a little bit of that portion on and spread them on a microslide, and then you fix it. You use the primary stain, which is crystal violet, to stain the first time, and then you add the iodine to wash it and make sure the iodine is reacting with the crystal violet. And then you decolorize. Decolorize is wash it out with alcohol or acetone. And once it's washed out, you're gonna stain it with safranin. And what it will give you is two different colors of stain. The gram-negative bacteria is defined this way. If they show a purple or bluish color, they're defined as gram-positive. If they show a pink or red color, they're defined as gram-negative. Okay, just like this show here. And the reason why they show different color, can any one of you tell me?
Because of the TQL.
Yes, very good. So, little bit, right? You guys learned microbiology already. So gram-positive, they have really thick cell wall, and those cell wall can restrain the primary stain and will stay there and remain a blue or purple color. Gram-negative ones, they have really thin layer. I marked here. They call it murin. It's the same thing as cell wall, okay? And the thing is also called peptoglycan. Anyway, so it's really thin and it won't be able to stay in the cell wall. So once you wash it off, it just comes like all off, and then it will take up the secondary stain and remain a red color. And if your bacteria has no cell wall, or it will not stain very well, it will show different variety of color or even no color, not so much color in them. So here, this is kind of important. I think all of the organisms on this list, you might need to remember their gram stain results. But I won't ask you to do it right now today, because you're going to learn every single one of them in the future, right? You're going to spend a whole year learning all of these. One thing I do want to mention that gram stain will give you a lot of information about the microorganism themselves, right? They will show different color for sure, and they will show different shape, and the shape and how they present themselves. Like for example, gram-positive, and if you got these gram-positive outside in a sputum sample, then you're highly suspicious for the staph or strep. And how to differentiate staph and strep in these samples? It's a lot of time the strep samples will show a binocular shape. We also call it double cocci, and meaning two cocci will stick together and make a double together. So that's their typical characteristic. And for the Staph aureus, they tend to stick together in a cluster, like a grape thing. So after observing your gram stain, sometimes laboratory scientists would have a brief idea of what I'm handling with already, okay? So that is very important. And here, if you have gram-negative, a lot of times the rods are more important for gram-negative microbes. And the ones that's not listed here, the curve, and you can read this list later on and make sure you're familiar with this. I won't go into very detail about every single one of them. So the use of gram stain is kind of a debate, like what sample should have gram stain. A lot of time, it is the very first step of test in the microlab, and it will return the result in a couple of minutes. And it gives you a quite clear idea of what you, the possible range of what organisms you're handling, especially when you're dealing with certain type of sample. And the gram stain will provide a quick preliminary result for the lab scientist and the physician, but it won't be able to identify what is the exact organism. They all could be toxin, right? Gram-positive toxin or gram-negative rods. But by looking at gram stain, it will provide a certain amount of information and give you a clue about what I'm handling or what I'm probably handling, right? So if you see gram-negative rods in a sputum sample, and especially if they have the encapsulated feature on the gram stain, a lot of time it is Klebsiella pneumoniae, right? You have this ping in your head. Oh, probably that. Then what do I need to do? I need to organize all of my biochemical tests and my selected media for my organism. And by looking at the gram stain results, actually it provides an opportunity to adjust your usage of antibiotic. Like I gave you an example. Just think of, previously when I see a patient, not me, physicians or pharmacists, they see a patient with lung infection, they may think, oh, staph, right? Or strep. But later on when they see, hey, it's actually gram-negative rods, no, they're not going to be staph or strep, then they might change their beginning antibiotic from erythromycin to this one, that one, there. So that's a idea how gram stain can help for you guys, for pharmacists. Other staining tests. We have tons of staining tests in the laboratory, but I do want to introduce these two just as an example for fungus identification and parasitic infection identification. So the Indian ink stain, it is the negative contrasting stain. So it's very helpful to identify the cryptococcus. And the giemsa stain is actually used, it's actually the base of the blood cell differential. Okay, so it's called giemsa stain. So with giemsa stain, you're gonna see the parasitic if they show in your bloodstream. For example, Plasmodium or Borrelia, or these kind of parasitic. They show there, they can show in your giemsa stain. Okay, so talk about direct observation. We know for sure, we even see those. I forgot to ask you one question about gram stain. So tell me if you think it's necessary to do gram stain on the stool sample. Like if you receive a stool sample from a diarrhea patient, are you gonna do gram stain on the sample? Using the sample? No? Why?
Yes.
Yes. Why? Because I asked why. Yeah, give me an explanation. What do you think?
So we can see what type of bacteria it is.
So do I hear no? You said yes, right? So there's a lot of bacteria.
Yeah,
Yes, it's too many. It's all kinds of crap in there. So basically we don't really do, because it's useless information. We probably can see the constitutive organism, but still it's a lot of interaction interference.
So it must be solid.
What do you mean solid?
Solid fecal.
A solid liquid, no matter what, if it's a fecal sample, we don't do gram
Okay.
stain.
So no fecal separate.
We do handle fecal samples. A lot of time, fecal samples sent down for molecular testing and streaking, like culture, not for gram stain.
Okay.
And a lot of time, you do examine the sample for parasitic, infection, eggs and stuff, and the parasites. Okay, so culture. Here, I list all these agriculture, but I do want to bring it to your mind. We have so many different culture media in the lab, okay? Depending on what organisms you're handling, we use different culture systems. But most often seen and presented are the blood agar media, because we begin with the sheep blood agar. This provides nutrition to support almost everything to grow. Okay, so these called nutritive media, like sheep blood agar or bovine blood agar. Sheep blood agar can also serve as differential media. What does that mean? It means something grown on it won't show the same characteristic. Okay, so here is an example. Different organisms grow on the same sheep blood agar gave you different presence. So strep pneumo will show alpha hemolysis, that's showing here. Alpha meaning you see a tiny zone of hemolysis. Hemolysis meaning you're gonna see a clear zone around your organism or your colony, okay? Because it's utilizing the heme inside the agar plate. So it's gonna clear out the agar. It's gonna be transparent, you can see through. So alpha hemolysis meaning tiny zone. You're gonna have a little bit hemolysis. And the Staph aureus is gonna give you a very wide zone of hemolysis that's called beta hemolysis. And some other organisms, mainly like Enterococcus, they will give you gamma hemolysis, meaning there's no hemolysis at all, even though they can grow on there, but you won't see hemolysis. So this is a way to differentiate. So we also have a little chart that you can compare. Like if I have alpha hemolysis, it's going to direct me to several organisms. If I have gamma hemolysis, it's going to direct me to several. So all of these are characteristics contribute to the identification of the organism. Enrichment media, I gave you an example, chocolate agar. It's because you added something on the agar to make it chocolatey color. So this is to help you to grow fastidious organisms, for example, Haemophilus, okay? Sometimes this sheep blood agar won't be enough to grow a lot of organisms, so you're gonna add something extra to support the fastidious. Enrichment broths, sometimes it's used for the detection of anaerobes, like TSV and stuff. So going on to selection of culture media and selected media. After you inoculate the sheep blood agar, you're gonna have different colonies, and the ones I show you, they have pure culture actually. But for your actual sample, you may have different culture, different clonings growing on your sheep blood agar. And later on, you need to pick your colony and transfer them into the selected media to give you more information. And one example is the McClunky agar. This only allows the gram-negative organisms to grow on them because they have the crystal violet to inhibit gram-positive organisms to grow. So if you have something growing on McClunky agar, that gives you a clue what? Yes, they're gram-negative, right? I don't know, rat or oxide. Anyway, Another one is the Columbia acid agar, CNA as another name. It will select the gram-positive organism and inhibit the gram-negative. So a lot of time you're gonna scrape both agars, right, with the same unknown. And then which one grows gave you, hey, this is that one. But this is one use of McConkey. McConkey is actually heavily used in the laboratory because it can serve as a differential media as well, not only selective. How to differentiate? McConkey agar allow gram-negative organisms to grow on them, but it will give you a pinkish, right, really bright pink color if your organism is a lactose fermenter, like the picture showed here. Your E. coli here, not a dangerous species, but this E. coli strain is a lactose fermenter, so it utilizes the agar substrate and make a bright pink color. But if you have a non-lactose fermenter, then you don't have a color change. So this second picture here, Pseudomonas, is the example for that group. So we use all of these other media or broth media to do supportive growth, to do differential growth, and selective growth. So based on our organism, whether they can grow and how they appear on these media, they also contribute to the identification of the organism. So culture, the culture itself, it's gonna grow and isolate some of these. I didn't mention fungi and viruses because we do grow and isolate fungi and viruses in the lab, but it's less presented and it's more complicated. And they do some of them, but not all of them. And as you can tell why. And you're gonna use tremendous amount of various media, and you're gonna go through the little chart to identify pathogen, and sometimes some of the strings, they show completely different growth on the same medium, right? And the pros is you're gonna get your pure culture, and this is very specific. You don't need to have doubts, right? This is growing, and this is the causative. The cons is it really takes time. Sometimes the fastidious ones take more than 2-3 days to grow, and that's why the physician constantly call the lab, where is my identification? Oh, I don't have it because it's anaerobic. It takes a week to grow, or it's a fungi, it takes really long to grow. And a lot of time you're gonna collect your normal flora, and they are gonna grow like crazy on your plate agar. And that's gonna interfere your identification as well, right? You have to be very familiar and knowledgeable with when you're handling the identification with different colonies. And of course, it won't be able to grow fastidious organisms, and not to mention anaerobics and stuff. So how to deal with that is to have other lab tests. Well, this right now I'm introducing is the biomedical testing. These always based on the culture. Right? You have to have the culture first, and you're going to use the culture to do all of these testing. So the motility testing is to see if your organism has like flagella, they can move around. Well, we have tons of these biomedical testing. I will just introduce a couple so that you have an idea. The indole test is to test out whether they can digest and produce indole, and if they do, they have the enzyme to do that. And the catalase is the organism, if they can hydrolyze the H2O2, then they produce a catalase, then they will produce the positive results for this test. And same thing with urease, not exact the same thing, it's just meaning the organisms, they can produce urease and utilize the amino acids. And later on, we'll have the color change as indicative of the urease production. So we have a lot of these, and actually close to 100. So how do you do that? If you do every single one by hand, it's taking forever, okay? Nowhere you can finish your patient with nowadays laboratory volume.
So they do have the commercialized, little test kit. You just need to make a culture suspension and add your culture suspension into each little valve, and then incubate this test kit for like 18 to 24 hours, and then it will give you the color change or give you any other indicators whether the test is positive. And all of these are different biochemical test results. And based on all of these test results, together with the culture presence, together with the gram stain result, you're gonna go through our little identification chart based on the information you're gonna go through. If I have a gram positive, and then my catalase is positive, and it's strictly aerobic, then I have micrococcus identified. For example, I see some gram-negative rods or bacilli, then my oxidase is positive but not curved, the shape is not curved, then I have Pseudomonas. Something like that, okay? Depends on how complicated your system is. So this is very traditional laboratory test way to identify, to make the identification. Nowadays, we jump into immunological tests and molecular tests a lot. So we heavily depending on this. So immunological tests, you guys all learn immunology, right? So what does that use is to use your immune system, the antibodies you produce or the antigens in your system that's going to trigger the immune response, make a detection. So if you have encountered a pathogen and they have triggered your immune system, what are you going to do? What are you going to have? Antibodies, right? No? Everybody fall asleep? So, I just want to introduce a little bit, background of the immunological test. I won't let you memorize all of these names, okay? That's insane. But when you have infection, your immune system is going to kick in, right? So your immune system here, what are you going to produce? You're going to produce antibodies, right? And your antibodies against what? Antigens. Antigens from where? Pathogens, right? Make a look. Anyway, so all of these will be in your system. So we got a tube of blood or something else. I won't complicate the situation. A tube of blood. So within the blood, I'm supposed to have antibodies, right? If you have the infection already. And I probably have the antigen depending on the phase of your infection, right? So all of these tests are developed to detect these things. Whether I'm aiming at finding the antigen, I have a whole bunch of tests to do that. If I'm aiming at finding the antibody, I have a whole bunch of tests to do that as well. And the most important one I do want you to be very familiar with is the ELISA, and you better remember the name. You're going to hear it a lot. So enzyme-linked immunosorbent assay, and I gave you several examples here, so you might be able to understand it a little bit better. But anyway, it's a test between antigen and antibody, depending on your target. Okay, what am I testing? Then I will have commercialized, or manufacturer will make commercialized counterpart to it, right? If I'm detecting your patient's antibody, then you're gonna buy antigen from your commercialized company, and those antigen will detect the antibody. But it also has something in between sometimes to make it more more sensitive. Okay, make the test more sensitive or more easier to carry out. But anyway, I don't want to explain this into details, but you do need to know the ELISA, and we use all of these immunological tests to detect either antibodies or antigens in your system, and all of these are associated with infection, right? And a lot of these actually are automated system, so even the lab techs or laboratory scientists won't really go into the details to figure out what I'm doing. You just load the sample and the machine is doing everything for you. So not even you guys need to know, okay? But anyway, so this is what has been basically telling you the immunological tests in the laboratory. Another use of the immunological test is the rapid diagnostic methods. Actually, these are quite oftenly presented them to the public. If you guys can give me an example that's very, very commonly mentioned nowadays. Huh?
COVID, rapid test?
Yes, COVID has tons of these, okay? FDA has cleared out like hundreds of the rapid tests for COVID, and they are all antigen or antibody-based immunological tests. And this is how easy they are, okay? But anyway, the reason for us to have rapid tests is because we want to make sure the test result has a really good turnaround time. We can return a brief idea, preliminary results to our physician or care team as soon as possible. So these tests are more designed for clinically heavily interested organisms like influenza, COVID, and C. diff, or mycobacterium. So other things, if they're not that, crucial for turnaround time, you won't really use rapid diagnostic because they have relatively low sensitivity and specificity. Molecular methods. I do want you guys to understand, I think you probably understand, what is the target that's been tested for molecular methods. That is your nucleic acid, not yours, your pathogens, right? So if I'm telling you, I'm doing a PCR on COVID-19, so I'm actually detecting the genetic information or the nucleic acid of the COVID-19, right? And it applies to all other pathogens, bacterial, fungi, viruses, but mainly molecular methods are used for virus detection. And there are rapid, not that rapid within 15 minutes, but relatively quick. So it really has a good turnaround time. And all of these, I do list a lot of names for you guys to get familiar with, okay? Not for you to memorize or what. So you can do the DNA probe, you can do the sequencing, you can do the PCR, and can anyone tell me what is a qPCR? Okay, so PCR, you guys know what is PCR, right?
Do you mind explaining? Sorry.
Oh, polymerase chain reaction is to amplify your nucleic acid in target, and so you can detect it later in a much higher volume of your nucleic acid. No, what is nucleic acid?
This one makes it.
Yes. So depending on your target, you could have RNA, you could have DNA, right? But anyway, they are detecting your genetic, not yours, sorry, I keep using yours, the pathogen's genetic information, the nucleic acid. So the qPCR is quantitative PCR. It's actually gonna give you the detection in real time, okay?
So for studying purposes, you want us to know each, describe each.
I want you to get familiar with each. I'm not expecting you to remember the name and understand how you can use it. I do want you to understand like what you're targeting when you're talking about molecular testing. Okay? So the one that's underlined actually is the one. Okay, so I also want you to make sure that you understand your target have different nucleic acid or genetic information. Like when you're dealing with COVID, you're actually detecting RNA, right? you're dealing with bacteria, you're detecting DNA. Okay, so this is one thing I do want you guys to know what is happening. I think it's also in your textbook. That is called multi-top mass spectrometry. So this is a trending technology in the microbiology lab, and actually it's going to take place all of the previous introduced stuff, to my opinion. In a short time, not right now. But anyway, the full name you do need to know. Okay, matrix assisted laser desorption ionization time of flight. Fancy name, mass spectrometry. So what it does is actually vaporize your bacterial samples. So right now it's mainly used for bacterial identification. So to begin with, you do have to have a culture. Okay, so you can't just load your sample on there. You have to have a culture. So on your culture, you can pick several colonies because you're not sure whether they're pure culture, they're the same colonies, or they're different. You can take several colonies that may be presenting different characteristics, and then you put onto the matrix and let it dry. And after that, you're gonna load it onto the multi-top. That's a machine, really expensive machine. But after that, the machine will take care of everything. It will actually use a laser beam to vaporize your sample and make charges, put charges onto your sample. And based on the identification of your organisms, they will take different charges and combine with different proteins. And when they move within the mass analyzer, right here, showing in the little picture in the corner, they will move in different direction and different speed. Okay, and eventually they will hit the ion detector on top. When they do that, they will give you a very different, like a fingerprint thing on the ion detector for each individual organism. So if there are different microorganisms, they will show different characteristics of mass spectrometry. So that's how your machine is going to detect and report the result. And based on a cycle fingerprint, so based on their database within the machine, they will give you an identification, which one fits your fingerprint. And a lot of time you're going to get a result. This is not in my database, which is okay. But most of the time actually within the database. And this can actually give you an identification more accurate and more specific than all of the other biochemistry tests. And even though I said the machine is quite costly, but if you consider the volume the machine can handle, it's actually very cost effective. I think right now at FinTop they are doing $5 each. Only cost $5 each. But if you're doing biochemical testing for identification, it takes 2 to 3 days and at least near $100 for each identification. So you can see the difference. Although you do need to investigate in the machine, the analyzer, and you do have to have certain type of people working on the machine. Not everybody is really trained to work with complicated or complex machine. But anyway, so for you guys to take away is understand what is being tested with multi-top and the name of the testing and the mechanisms of the testing. Okay, that's also in your textbook, I think. So that's the part for testing. So testing, I'm almost done. There are a lot of testing I didn't introduce because it's really a lot in the laboratory, and you guys won't be able to really take away those information anyway. So you will be exposed to them later on when you go in depth with your ID study, right? So next thing, and I think it's the most important thing for you guys for today, is to understand the in vitro antimicrobial susceptibility testing, right? That's also done in the laboratory. And this is to determine which antibiotics are useful for treatment. So we do know what is causing the infection and how can we treat it. Can we use any type of antibiotics? No, right? To make it more efficient and safe for the patient, you have to choose. And all of these testing here has to be, well, most of them fall into the standard of Clinical and Laboratory Standard Institute. They have certain type of procedure for you to do that. So to begin with, this is a qualitative testing. So qualitative meaning it won't give you a number, but it will give you a status, whether it's susceptible, it's resistant, or intermediate. So this diffusion, also called Kirby-Bauer method. Kirby-Bauer is quite a laboratory guy. He has named several lab tests in clinical laboratory. But anyway, this diffusion is how you do that. You actually strip a plate with certain standard suspension, okay? Not any concentration, a standard suspension. So you know what is the concentration of your bacteria on the plate, okay? That has to fall into that range. And you're gonna place your little disc, paper disc, that is saturated with your antibiotics, and place them onto the outer plate, okay? And you incubate the outer plate for a certain amount of time under a proper condition, temperature. And later on, after the incubation, you're gonna take it out and measure the zone of inhibition. And what is zone of inhibition is the clear zone, okay? If you can see here, around the disc, there is a clear zone. That means no organisms been growing over there, right? That means they are inhibited by the antibiotics. So that's the zone of inhibition. And you're gonna compare that zone, the size of the zone, the diameter of the zone, to your standard zone size. So with each paper disc, you can buy them from manufacturer. They have a standardized zone size to give you as a reference. And once you compare them, you will know whether it's considered susceptible, resistant, and intermediate, which means I don't know, right? How to interpret? The larger the zone, the more sensitive is the organism to the specific antibiotic, right? That's easy to understand. So what do you report? Like which drug do you select? Like if I have tested out 6 antibiotics here, and they gave me this result. I'm telling you, this one is susceptible, that one is resistant, so which one do I choose to use on my patient to treat the infection? Yes, you select the one that's at least susceptible outside, right? I don't really know what's gonna happen inside, but at least you can use the resistant one because outside it won't kill the bug, right? And this is actually a useful way to screen for multiple organisms or colony types, like showing in this picture. If you can see, this is a zone of inhibition, and here as well, but you do have several colonies growing here. That gives you an indication. Your culture is not pure culture. You have different organisms within the same culture. It's a mixture, right? And some of them are inhibited by this drug. Some of them are not, right? It's falling into the resistant zone. So this is the indicator. So you need to further work out this culture. Isolate more. Anyway, I do give you an example of intermediate, that intermediate, meaning following this zone, you cannot really use it. It's in between susceptible and resistant. So based on the drug and organism, they do have different cutoffs or breaking points for these interpretations. So another testing method actually do want to introduce is the semi-quantitative. So it begins with broth solution. So all of the susceptibility actually begin with broth solution. What you do is you're gonna make a standard or series solution of your bacteria, okay? Not your bacteria, make a serial solution of your antibiotic drugs. And you're gonna add each concentration into your broth culture of bacteria. And the one actually inhibited the growth of the bacteria. The first one inhibited the growth of the bacteria, would consider the minimum inhibitory concentration. So for example, this one is a serial dilution from 8 microgram per milliliter to 4 to 2 to 1 to 0.5 to nothing, right? So within this 6-2, the antibiotic concentration declines, right? And with the same microorganisms growing in the broth, actually this one is the lowest concentration that can prevent a visibly growth of your bacteria. So that is considered the minimum inhibitory concentration. So this actually is specific to each antibiotic and each organism. So it's going to be different for each. And you cannot compare these among different antibiotics. And for you to do the broth solution, you also have a standard inoculum of bacteria. So you have to make sure all of the tubes have the same concentration of bacteria to begin with, right? Yes, so for my example, the tube number 4, or the concentration of 2, would be the one that's considered MIC, right? MIC is the most important thing you need to take away today. So the susceptibility breakpoint actually is established beforehand, before you're doing it. So you're gonna, or the clinical and laboratory standard institute, they're gonna do a bunch of testing on hundreds of microorganisms, and they will establish the reference inhibitor zone size breakpoint. So meaning they're gonna do the agar this diffusion and decide which is the zone breakpoint, the inhibitor zone. And they'll also correlate the same concentration with the liquid broth and to decide which is the MIC for that inhibition. And they will plot it using a chart, and then use the plotted chart, you can find out your breakpoint for each. Okay, so this is an example for a report for susceptibility. Take the piperacillin as an example. So you have a reference cutoff or breakpoints for susceptibility and intermediate and resistance. So if your measured zone diameter is greater than 21 for that specific organism, then your organism is susceptible to this antibiotic. And if your zone of measurement is smaller than 14, then it's considered resistant. And it also relatively converted to the MIC breakpoints. So for any susceptible result, that meaning the MIC breakpoint is less than 16 with the units. And for anyone that is resistant, consider MIC breakpoint is greater than 128. So in the future, when you're reading the lab reports, they will show you the reference range for each test result, and they will certainly give you a answer whether it's susceptible or resistant. And you won't be able to use the one that shows that intermediate or resistant. So based on the MIC and the broth dilution method, we actually have automated antimicrobial susceptibility testing system developed. and actually use a micro well system. So you're basically doing the zero dilution within microplates, right? This is one of the microplates you can find on the market, and they do have different systems as well. And within this microplates, you're going to do a zero dilution or doubling of your antibiotic reagents, add it on to the pure culture you collected for your microorganisms. And the first one, or the lowest concentration that has a clear well, meaning no growth within your system, within your treatment, will be considered MIC for your testing. So this way, you're actually doing multiple drugs at multiple levels of concentration at the same time. Okay, so all of these is about to save you manual work and also make sure it's standardized and comparable. So the mechanisms like this, we do have several available choices on the market. Depending on where you're going to go to clinical rotation, you may see different. Okay, so all of these use the same mechanism. They're going to do the microtiter, microplate at the background. They do use different mechanism to detect the growth. Okay, so how you can tell there is a growth? Us, we look at the tube. If it's turbid, then we say there is a visible growth. But for the machine, they use different mechanism to detect the growth. But basically, how they define which antibiotic is useful or susceptible is based on the same mechanism. And this is one example for a lab report you're going to get from one of these automated systems. So what you're going to get from here is to understand how you can use MIC as a method to define which antibiotics is susceptible and which is resistant.
So when it's equal or greater than 32 is resistance.
Right, that's an example. So it's based on a pre-developed system. So normally the clinical standard institute, they will have a pre-established, and all of the commercialized systems, they have their standard for each individual drugs and individual organisms. So they will have a pre-established system for you to compare with. So what I gave you is just an example, okay? What time is it?
14.
Oh, you guys need to take 5 minutes break?
It's gonna prolong class.
Okay, so one last thing for this part is just to introduce other susceptibility testing. Gradient strip test is one that's kind of in debate. Okay, I'll tell you, it's actually very efficient. It's a combination of the disk diffusion and MIC. So it's gonna give you a number, whether like right here. When you read this, you apply the disk just like the little one, the round one, but it's gonna be a strip, right? And the strip is gonna produce elliptical zone of inhibition. And within the end point right here, it's gonna give you a reading of MIC. What is exactly the MIC, the break point for inhibition? So it's very useful and easy to use. The only concern is it's very expensive. That's why it's less used nowadays, because there are a lot of easy ways to get around. I do want to put this into your knowledge bank. So in the future, if you see this, when you go to smaller labs, not nowadays, like a lot of the central labs, they are highly automated, so these kind of tests kind of faded out. But if you go to a rural area, smaller lab, they may still be using this. So you won't be surprised when you see this. Okay, so this is actually a combination of this diffusion and agar diffusion. I do want to talk a little bit about superbugs, the bugs that can be resistant to certain type of antibiotics, and how to detect their superbug nature. And normally it's just by detecting the resistance factor. Okay, one factor that we want to mention is the beta-lactamase production. So if your organism has the ability to produce beta-lactamase, what drug is going to be resistant to that? What? Yes. Easy, right? Yes. Or that family,
Yes.
penicillin family. Trust me, I don't know all the names, right? So. Anyway, so one test we use quite often is nitrosamine disc to help us to detect the ability to produce beta-lactamase. So this is actually going to give you a color change if you do have a bug that's producing the beta-lactamase. So if it turns from yellow to pink, meaning your bug is kind of resistant to penicillin or it's the penicillin family. So you need to think of alternative. And it's quite useful in the assessment of staphy, cocci, enterococci, and all of the others. But it won't be able to give you an idea the beta-lactam resistance because they have certain type of altered penicillin binding proteins. But anyway, so this is one of that's still used in the lab. Another one is called chromogenic agar technology, and within this agar, you normally have chromogenic substrates plus cephalosporin. And the cephalosporin will prevent some growth of other organisms, but not the MRSA. Methicillin resistant in Staphylococcus aureus. So if you do have a MRSA, then it should be able to grow on the plate, and then once it grows on the plate, it will utilize the substrate and make a color change. Okay? So here you will get a different color, and it could be this color, it could be even brighter pink color depending on what substrate your agar plate is using. So this method is actually used for detecting the ESBL and some of the Enterobacteriaceae. But to be honest with you, I think it's kind of faded out by molecular tests because nowadays we use more often the molecular test to detect those genes in the microorganism, right? So now you need to remember what genes in charge of what. So the MCA gene is in charge of what? If you do have that in your staff, what do you have? If your staff is positive for that gene, what it becomes? MRSA, right? So that's why I'm saying it's fading out because MRSA can be detected by, if you find this gene, it's MRSA. And the same thing for this one, enterococci, if you find the bank A gene in your enterococci, then you have vancomycin resistant enterococci. Okay? So that's two things you need to remember, I think. But I think you will revisit the topic once you actually learn those microorganisms. Okay, so last part of today's lecture would be a little bit drug monitoring. We do that in the laboratory, and you guys probably understand the logic why we're doing that because some of the drugs can be toxic to patients, right? You want to make sure they're killing the bugs, not the patient. So all of that reasons, and the testing method here we're going to normally use would be the fluorescence, polarization, immunoassay, or the ELISA, enzyme-linked immunosorbent assay. Sometimes certain type of drugs, they use HPLC as well. So just to throw in names so that you will be familiar with those. And I do want to mention the two drugs that's constantly monitored in the lab within the patient serum, that is vancomycin for sure, and if I can say the name correctly, aminoglycoside. Is that right?
Yeah, you did good.
Good. Yes, those two types of drugs are constantly monitored in the laboratory using these methods. Let me see if I have. Okay, so it's time to review some of the concepts we talked about today. Okay. I just want to enhance that I do highlight, and all the things that I highlighted are considered important to my class. So you know where you need to focus, right? The gram stain and antibacterial susceptibility test. Okay, do I have a volunteer to read out loud to the group? Anyone wants to read and answer the question?
Choose the correct statement regarding the white blood count in differential. A. White blood count is usually elevated in response to infection. Is it B? The normal range of the white blood count is 12,500 to 20,000 cells, whatever MM3 is.
Milliliter?
What
That's
is
the same as milliliter.
Oh, okay. Or is it C? White blood count is not specific and can be elevated in response to a number of non-infectious conditions. Or is it D? Neutrophils are the most important type of white blood counts in the blood.
Okay, do I have an answer?
E.
I heard all of them. Unfortunately, that's not.
A is wrong. A is right.
It's right.
Well, it
Is
does
that
usually.
Yeah.
So if there's no usually, I would say wrong. But it says usually, so I'll give it. Right?
I thought white blood count was down.
No, if it's a bacterial infection, normally you will have increase. Leukocytosis. Remember?
Okay.
Okay, so a slightly decrease happened to viral infection.
Okay,
Sometimes,
I'm
not all the
thinking
time.
different.
Okay?
Okay, this is bacteria.
And neutrophils are the most common type, that's no doubt. Okay?
Yeah, we talked about that.
And C is also correct. White cell count is non-specific, and a lot of things can increase your white cell count. That's why I introduced as biomarker, right? Only thing is wrong is B. That is way higher than our normal range. Go back to revisit the normal range. Even though I told you it's gonna change among different hospitals, different labs, but it do have a certain range.
So what count would you want?
It's at the very beginning.
Oh, you don't have to show it.
It's 4.5 to 10 to the 9 power.
Okay.
Yeah. Okay, so that's your correct answer. And number 2. Do you guys want to read it out loud, or you want to spend time reading it and getting the answer?
We can give you answers.
Do I have an answer?
All of the above, right? All of the
I'm
above.
going to need you to sit right next to me during this test. Yes.
I
I figured that one, yeah.
I I'm sorry to say that, it's not.
No, these not, that's not in a few hours.
Yeah.
It takes
You
days.
said it takes days.
Yeah, even
Yeah.
a
That's
week.
wrong.
I remember you said doctors always like.
But you said it's not yet possible.
But it's,
You said not yet possible.
Not yet possible with where?
At the bottom.
At the
At the bottom.
D, answer choice D.
So that's where we, so that answers.
Cause you said it takes days.
Oh. But it's just in general, it could be because you can use molecular testing and immunoassay, right? So that could be possible. And also the money talk, it can be as short as 12 hours.
Yeah, that was rapid. Yeah.
Okay, Monitab is not targeting the genetic markers. It's the cultures, the cells themselves. Okay, the organism.
We did a speed.
So
And B, cultures can provide definitive pathogen identification and differentiate organisms on the bias of more rapidly. No, that is on exact the opposite, right? Molecular base is way faster than cultures. Cultures take days to grow,
yeah.
and you have to do all of the biomarker testing and all of that. It takes really long time. Okay.
Sorry. See, you said it was wrong.
C is wrong because it's targeting the organism itself, not the genetic markers. It is using the organism to make the footprint, the fingerprint, to make their ways different presentation in the mass spectrum.
Okay.
Okay? The genetic marker has been analyzed by molecular methods.
Okay.
Okay, MSC. This should be easy, right? Which one? C. Did I get C? Perfect. Okay, so that is the most important thing to remember. Okay, number 4, methicillin resistant Staphylococcus aureus is detected using agar containing.
This is for sure you will know.
Oh, the 10-day.
I don't know any questions. I just said from
This is
as
Marcia.
a clinician, I would say you definitely will know this because I see this culture almost every day when I have my clinical duties. I always see this. You definitely need to know.
I think it's C. C is C.
Oxycillin. Okay, and sodium chloride.
Remember that, okay? I'll keep testing you.
Okay, last one. Which of the following, what?
Can we go to that slide that regards to? I don't understand that.
Which slide?
That can help us. with
Marxism?
that. It's that one.
Super.
Where does it say it?
They don't say.
You should remember it.
Yeah, I was trying to figure out where does it say that.
Oh, it's in your textbook.
Oh. See, I didn't read my textbook.
Yeah, I
before
didn't
clicked twice.
write it down here, but it's in
Okay,
your
but this
textbook.
needs must.
Yes, that is important to remember.
What test do you though? So you remember, right?
Okay, the last one. Following technology is not currently used for rapid. What do I hear?
Okay,
Some kind of tricky question, huh? Not currently used.
Not currently.
oh, not currently. Okay.
So it depends on how rapid they are, right? They are all kind of rapid.
Yeah, but
So-
this was like.
That's why you choose D. All of the above are used for rapid.
This
I
may
couldn't
become a
find
multiple
C.
choice question. Multiple answer question. This is a good multiple answer question.
Please, no select all that apply.
So because all three of them are used for rapid detection and rapid identification, okay?
But I didn't find that in the slide for C.
Enzyme linked, it also gives you rapid results.
Oh, that's part of the one
That's
that you
the
said, go look it
automated, yes.
up. Okay.
You hear that C is actually, we always said ELISA. It's abbreviation. Did you hear about that?
Yes.
Now you know? Yeah, I feel the same way. Sometimes I heard of this abbreviation so long. Sometimes my students say, what that abbreviation say? I don't know. I didn't Google it. Okay?
Yeah. So references, and I do want you guys to check out the introduction of microbiology lab if you have time. Okay, thank you very much.
Thank you.
Dr. Wang, for this lecture, do we have the book?
You can use that access. I can tell you which chapter. You have.
Because she keeps
You have,
referring.
all of you have access pharmacy, right?
Yes.
The Imperial book, infectious disease, the first chapter is this one.
Oh.
It's microbiology. Read that chapter, okay?
The
Imperial book, that's our textbook.
Okay.
When I start to review the syllabus, still have some time. So I will review the syllabus with you.
I need to stop recording. I forgot before I close it out.
Okay. Okay, so how many of you get a chance to read the syllabus? Are you worried? Scared? Okay. The top purpose on here is to help your study, to help you prepare yourself for the clinical practice for your P4 year, to get yourself ready for the NAPLEX. That's all the purpose. I'm here. I'm not here to get your challenge. Well, the pharmacy school is always challenging, but my purpose is not add more challenging to you, but my purpose is to help your study. That's all the purpose, okay? So, when you read those syllabus, you will see the textbook is actually the Imperial book. You can go to the Access Pharmacy and you can find that. Thank you. You can find that book here. There, actually, the infectious diseases section. Okay, the first chapter is microbiology or the lab test. Okay, because back then the pharmacist office, the infectious diseases pharmacist office is next to the lab test. Just next to the door was the lab test office. So we close work together. In hospital, the antibiotic stewardship committee always have the lab technicians, their director always join our meeting. All the pharmacists, ID physicians are in the antibiotic stewardship committee because we work together to develop the test. The new antibiotics coming to the market, how we need to develop the test. The CTSI developed their cutoff for the MICs, for all those bacteria. And then our lab need to work together with the pharmacy and the physicians to develop the new guideline for all the test for all the ID. So ID is microbiology is definitely foundation. So Wednesday I will give you more about the bugs, which I mean bacteria, and also how we select antibiotics in general concept. Okay. So this semester we will develop actually I took over the coordinator for this. This is new to me as well. So I actually reorganize the ID teaching because of my own experience in ID practice. I'm the infectious disease pharmacist at Houston Methodist Hospital. I see a lot of ID patients. And also I reviewed for our student, the P4 year, the NAPLEX, the ID section, I reviewed for our students since 2018. I really know what they test for the NAPLEX. So like I said, I will help you for your study. If you have any questions, feel free to contact me. So if you the questions for the entire class, so you have a rep, your rep can help you to address all the questions to me. I don't want you 60 students send me email a day, then it will like what? So if you have individual questions, feel free to email me. But if you have like the class lecture related question, ask your rep to email me so we can address it for you. And as you can see from the course, the lectures, we'll start from some foundations like lab tests, bugs and drugs. We will cover the beta-lactam and all the other antibiotics classes for a few classes before we start to talk the real infectious diseases, okay? So that's how we set up this class. But supposedly your pharmacology class, the PODA, before this class, you have the pharmacology, the PODA class. That class is supposed to give you more details about the pharmacology of each medications or classes, okay? So our class, before we start talking infectious disease, those beta-lactam or antibiotics you see, I will give you the review, but that will be, we only have certain classes to cover, so that will be more brief. But I want you to have antibiotics knowledge before you start real infectious diseases. Several times in the past year, you go to the older years, we don't have that. When we start to talk about the UTI, and some student was like, I don't know about the bacterium. Say, what? I didn't have that class. Show me the syllabus. Yes, they didn't have that class. So now that's why I set up some antibiotics review. But for more details about the pharmacology, you should go refer to your PODA, okay? Then we will talk about the real infectious diseases, starting different bacterial infection, which is probably half of the diseases I saw in my own clinical practice is bacterial infection. It's a lot of drugs there, so you need to spend a lot of time to remember those brand and generic. Every exam, we will have 2 bonus questions to cover the brand and generic, okay? Study hard. And then you have 5 integrate exam and 1 final cumulative exam. Those exam dates are decided by the university. All the P2 and P3 have integrated exam date. So your P2 actually had the integrated exams, so you're familiar with that, which is great. Also the grade.
To direct antimicrobial pharmacotherapy.
Okay, hold on.
Finally,
You want to review that after this class, right? Okay, so I need to go to the grade. So the grade, as you can see, I want you to study really hard on daily basis. I had classmates super smart when I was here, back then, 10 more years ago. So he always studied right before the exam, no sleep, and study all the way the entire night. And I don't. I study every day. I sleep. I just pretend no exam the next day. I just sleep. I got good sleep. I just study every day. So this will be, the syllabus is tailored for your daily study. I want you at least schedule some time, no matter how busy you are. You all have your personal life, but pharmacy school, you are here. You pay the tuition, you don't want to. In the future, you cannot pass NAPLEX. And then you cannot be a pharmacist, you cannot get to pay as a pharmacist. Then you will regret. You don't want that happen, right? I don't want that happen too. So you want to study every day, but you can see from here, we have 1 TBL, 5%, and 1 reflection. Definitely you want to do it. I will give you the instruction. Pretty much we'll do that before the mid-exam. So I want you to do some reflection to see what you can do better, what you already you think you did good, you want to improve. So keep doing some reflection. I will grade individually for all of your reflections, and that will 5%. And also I will give you weekly quiz. Those quiz will not like the real exam, like more like target very small key points. I will test you again and again, so you know you will still can recall when you sitting there for the NAPLEX, because the book is like this thick. You probably some of you already saw that RX prep book. So it will be super hard if you cannot remember those right now. You would like to remember many of those key points right now. So I'll keep testing you that. And also, you have total, like I said, 5 integrated exams, and each of them will be, actually I will drop one lowest exam for you, okay? So total will be 4 exams out of the 5 count toward your final grade, and each 15%. Fair? Okay, if you cannot come for one exam for some reason, we don't have makeup exam as well. Is that clear? Okay, so the cumulative will be 1 final cumulative. So for that one, because it will be the SHP mid-year time, I don't know how many of you want to go to the resident, and I definitely need to go for the mid-year that week, the last week of the cumulative exam time. But school will have the for the integrated exam, the cumulative exam, they will have the faculty proctor here. I will discuss with the school for those students who go to mid-year. Maybe you will come to my room to have your final exam. We'll see what we can do at that time. We'll see.
Yes. So
Yeah.
I was thinking, can we possibly, those who want to go, if we could do it on the 9th?
You have, can you do what?
Can we just take it on the 9th when we get back?
Which date was
That's a
that?
Friday. Because the last day is on a Thursday.
That's the cumulative exam, is all the exam dates. I don't think I can change it. It's because that is like second year and the third year has the same scheduled exam date.
It's a special circumstance.
But I know I will also go to the mid-year, not just one student. We have
Yeah.
big event,
Yeah.
so I will ask the school what we can do for that. That's not just for one of you.
If you go to your
But if for
faculty,
half your class, say 2/3 of your class, took on the 7th, then I have to make another exam for you in the 9th. That means I need to make 2 cumulative exams. But then some students will say, their exams are easier than mine.
oh no, we don't discuss that stuff like that.
So I don't know, but I will get you the answers. So come back to me at the beginning of November. So we'll discuss that. But I definitely need to go because I am the primary advisor for the SSHP. So I must go, and also we have some fellowship program. I definitely need to go there. So we'll see what we can do while the time getting closer. And then your both of your midterm and final, I give you remediation. I give you remediation opportunity. Both for the first, I would say the first 3 exams and the second 3 exams, you both have the remediation for 1 exam. One for the first 3, 1 for the last 3. Make sense? So those remediation will be specifically go to your remediation section. We follow up your P2 years, Dr. Pong, what she did. Okay, so that will be same as what she did for your class, because I probably you can see my syllabus is adopted from hers. So I respect her a lot. She's very good. So that's what we will do for this year. I look forward to have a productive year with you and help your study as well. You're welcome.
May you please give us your email address because the syllabus doesn't.
Oh, my contact information is in your blackboard. The faculty information is there. You can find my information there, my email, and my office phone number.